Thanks, Marco, for inviting me to this wonderful event. I'm thrilled to be here. My connection with Larry started many years ago when I was a postdoc at Oxford University, where I was supervised by Larry and by Alan. Larry was, I guess, a 3-year-in chair of the department and was very busy, and he delegated some of the supervision to Alan, and so I had the pleasure of working with both of them. There were 2 years that I spent there. I will say I came on Canadian money. I had a postdoctoral award from the Natural Sciences and Engineering Research Council, and I discovered that my $6,000 that I was paid every year, that the only person in the department who made more than me was Larry. So when I got a real job after that, in the UK, I had to take a cut in salary. But being a postdoc at Oxford was therefore a rewarding experience in many ways. I worked with this animal, the Syrian hamster, following on from some work by Jerry Schneider, and I will say that in the 2 years of work that I did on the hamster, nothing came of it. The greatest problem was not the anatomy or the neurosurgery or anything of that kind. It was that when we deprived them of food or water, they promptly hibernated. They went into a torpor state. So we found it very, very difficult to actually work with this animal. But despite that, I spent 24 months there learning a great deal from not only the people at Oxford, but also the people who came through. And one day, at the end of my tenure there, Larry came out to the garden at the back of the old building on South Parks Road and asked me if I'd like to move to Scotland. I don't know, he wasn't really just trying to get rid of me, but he really had a line on a job. And I ended up at the University of St. Andrews, where I stayed for 6 years as a lecturer, 6 wonderful years. Because I started a long-time collaboration that still goes on with my friend David Milner, who had come to St. Andrews a year before me. David and I applied and were very successful in getting a grant in 1973, but there was a proviso attached to that grant, and that was that we had to make a journey down to Oxford to talk to Larry, who was going to set us straight on what we had to do in order to be a success with that grant. I have to say that Larry was very gracious and said, well, I'm not quite sure how much advice I can give you, but I know you'll do a good job once you get started. And with that encouragement and indeed some practical advice, we started a long-time collaboration, David and I, and for that too, I'm grateful to Larry for setting in motion. I went back to Canada in '77, and I'm still there at the University of Western Ontario. And I'm going to start my talk by showing you this cartoon, which I think is a cartoon for the person in the street's view, and indeed some scientists' view of how vision works, that vision is in the business of painting a picture of the outside world, an internal representation, a simulacrum of the real thing that can be used for thought and can be used for acting on the world. Now, for many reasons that aren't worth going into, this can't be right. The most familiar being that what is the homunculus in the brain that's looking at that, and what's looking at the picture in the homunculus's brain, and so on, reductio ad absurdum. It can't be right. But even if one can't talk about some kind of realistic simulacrum of the world outside, one can talk about a psychological representation of the world. I mean, I have one. I see people out there. I see chairs. I have a psychological representation of the world. But even admitting that, one has to concede that vision didn't begin as a system for representing the world. Vision began as a system for the distal control of movement, and this is perhaps one of the simplest examples of this, a euglena. And euglena, as you know from your biology course that you took in high school, will move its flagellum a great deal when it's in the dark part of the pond, and when it's in the light part of the pond where there's a resource, sunlight, it will not move its flagellum very much, and you don't have, and as a consequence, the euglena will manufacture food through chloroplasts in its cytoplasm. Now, one doesn't have to argue that somewhere inside those little organelles or in that cytoplasm there is a representation of the world outside. You don't have to talk that way. You can talk instead about some kind of transformation of energy from light through photochemicals into movement in microtubules that actually produces contractions and changes in the flagellum, and that's really all you have to say. If you get to vertebrates, can you be equally mechanistic? Can you talk about vision without representation? And indeed, I think to some degree you can. Some beautiful work by a number of people, David Ingle in particular, has shown that there are at least 5 separate pathways from the eye to premotor neurons in the brainstem that control different classes of behavior. So, for example, there are projections that go from the eye to the optic tectum that are involved in the visual control of prey catching. There are projections that go from the eye to the pretectum that are involved in the visual control of locomotion around barriers. And the separation of these pathways is such that you can rewire one and produce an animal that behaves in a mirror image fashion with respect to stimuli in one class of behavior but not the other. So these are very independent pathways, and there is no need to invoke the notion of a general representation of the world in order to explain the behavior of these creatures. So, as David and I argued, vision evolved not then as a single system that allows organisms to see the world, but really as a kind of an expanding collection of fairly independent visual motor modules. But of course, the big question is, what about humans? Are we organized the same way? Well, if you look just at the projections from the eye in the spoxology diagram to the first synaptic contact with a structure in the brain, you can see there's at least a suggestion at the get-go that there's some independence in the pathways. I mean, we have projections that peel off fairly early, that go to the hypothalamus, the suprachiasmatic nucleus. They're important for synchronizing circadian rhythms. They're working overtime in my brain right now. I assure you, I flew here yesterday from North America, and it's all I can do to stay awake through my own talk. But and that's because these photosensitive cells in the retina are synchronizing the endogenously generated circadian rhythm in the suprachiasmatic nucleus and getting me back on track. There are projections to 3 sets of nuclei called the accessory optic tract, again in the midbrain, that are important for the visual control of posture. Their sensitivity, interestingly, for whole field movement across the retina correspond very nicely to the axes of the semicircular canals, so that the vestibular system and the visual system can work closely together for the control of movement. There are projections that we've just seen in the frog. They're important for the visual control of locomotion around barriers. There's structures like the colliculus that have been well studied that are important for saccades and fixation. But of course, there's a major projection in the human brain that goes to the dorsal part of the LGN, and then up to primary visual cortex, and then on to other cerebral areas that has been divided into the dorsal and ventral stream, and that's where most of the activity of cognitive neuroscience is focused, I would say, over the last 100 years. While one can talk about independent visual motor pathways in vertebrates, it's clear that as life becomes more complicated, particularly socially, we get this emergence of a perceptual representation that we can see certainly in humans and our primate cousins, but also probably in many other mammals as well, if not birds. And these general purpose representations really do have a big advantage in that they allow us to choose goals, to plan ahead, to, as it were, posit what might happen if we did this or did that, and communicate about all of this on the basis of the shared visual experience with other members of our species. And this clearly is important. But David and I would contend that while these visual representations allow us to do it, to actually choose a course of action, the these systems have no direct contact with the motor system, but have to go through various cognitive buffers. And as we'll see, there are other systems that are important for the direct control of movements that we make. Whoops, am I still there? Yes. At the same time, there was increasing emphasis in the primate, at least, on the generation of skilled movements, so that within the cerebral cortex, these more primitive visual motor channels that go from the eye directly to the midbrain and brainstem are modulated and controlled by cerebral structures, as we'll see, in the posterior parietal cortex and through their connections with premotor cortex and motor cortex as well. So I was in Western Ontario, David still at St. Andrews, and he and I, over a series of studies, postulated that there was evolutionary pressure then for the emergence of two kinds of visual systems. One system that allows you out there to recognize the cup and the pencil, the hand, the watch, the piece of paper, and so on, and to recognize there's a picture. It's not; those aren't real things. You know you're looking at a picture. But that visual system is functionally, and certainly in terms of the neural structures that it engages, very different from the visual structures that allow the actor in the picture to reach out and pick up the cup. That fundamentally different transformations are involved, and as I said, different neural pathways are engaged. And indeed, David and I suggested, as you know, that we could map this distinction between vision for perception and vision for action onto the two very prominent sets of pathways that have been identified, projecting from early visual areas up to the posterior parietal cortex on the one hand, and down to the temporal lobe, occipital temporal areas on the other. The dorsal and ventral stream, the dorsal action stream and the ventral perception stream. Now, over the years, we've accumulated a good deal of evidence for this account, but the starting point really, as some of you know, was this patient DF, whom we were fortunate enough to meet in 1990. Patient DF at the time was a young woman who had the misfortune to have a shower in a badly ventilated bathroom, and as a consequence developed carbon monoxide poisoning that left her with damage throughout her brain, but with a great deal of concentrated damage in area LO, the lateral occipital region, we now know is part of the ventral stream in humans, that is important for the recognition of the geometric shape or form of objects. Her most salient symptom is what is sometimes called apperceptive agnosia following Lissar, or visual form agnosia more recently. That is, she was unable to recognize objects on the basis of their form, although clearly she could recognize objects through color and the surfaces, the surface texture of objects. And I want to emphasize that clinical and psychophysical testing was pretty well normal, in the normal range. So her deficit was so profound that when David first encountered her and held up a pencil in front of her, she did say it was yellow, but she couldn't tell him whether he was holding it upright or horizontally or on a slant. But when she reached out to pick up the pencil, remarkably, her hand was oriented in the correct orientation, despite the fact that she couldn't report that. So that distinction between her preserved vision for action and her clearly deficient vision for form led us to carry out an experiment many years ago, which is a version of the pencil experiment, really, in which we asked her to take a handheld card and to match the orientation of a slot placed in front of her. And when she attempted to do this, what we've done here is to rotate the correct orientation to vertical, and what you can see is that she's terrible, obviously, compared to a control subject. But when you change the task and you ask her now, look, I know you can't see the orientation, but just, trust me, try and post the card into the slot, then remarkably, she tunes up beautifully and she's able to do so. So it looks as though, despite the fact that she's got damaged form vision, somehow her motor system has access to information about the orientation of the slot so that it can control the orientation of her wrist as she reaches out to post it. So, of course, DF had damage to the ventral stream. What about patients with lesions of the dorsal stream? Are they the mirror image, as it were, of DS performance? Well, as it happens, 2 years earlier, Marie-Thérèse Perronin had tested clinically, in a more clinical version of the slot test, a series of patients who had posterior parietal lesions unilaterally. And what she found is that when you presented that slot in the field contralateral to the lesion that left them with optic ataxia, they were unable to orient their hand to the orientation of the slot until they made contact. And then they could use haptics to put their hand through the slot. So, you know, it was an important double dissociation then that we had neuropsychologically. Of course, all this was going on while fMRI was not on the scene at all. What I did to create this graph, fMRI took off very quickly from 1992 when there were 2 papers published. And I created this graph, which is a huge underestimate, by doing a Boolean search of PubMed, by putting in, you know, fMRI, MRI with functional, and so on and so forth, until I counted up the papers. I gave up after 2010 because I was running out of space on the PowerPoint, and I wasn't ready to rescale it. But we were studying DF back here when fMRI had not yet been discovered. One of the postdocs who's on both of those initial papers is Ravi Menon, who worked with Goodale on this. And Ravi we hired him in 1994, 2 years later. He's still with us, and he directs our Center for Functional and Metabolic Mapping. We have a 9.4, a 7, and a 3T. We have 3 3Ts. And through Ravi's astute stewardship, this has been a real successful imaging center. And one of my postdocs, who's now a full professor at Western, Jody Cullum, led the charge on actually scanning patients like DF to see if we could find fMRI evidence that was coincident with the findings that we had so many years ago in neuropsychology. This is heroic work, not only for Jody and the experimenters, but also for the subject, because you have to have people looking directly at the object they're grasping, which means you can't use mirrors. So you tilt the head, and you have to, therefore, through Ravi's help, develop a coil that will work efficiently for actually scanning the brain while they're doing this. You have to have the upper arm fixed, and you can present stimuli on a grasparatus, this rotating thing you can see here, where you can back illuminate a plexiglass object, and they can grasp it in the dark. And when you do this, what you find is that in DF's brain, this is DF's brain shown here, the lesion in LO is marked in gray. What you can see is activation in both hemispheres in an area that was identified at the anterior end of the intraparietal sulcus that corresponds to what you see in control subjects when she reached out to grasp objects. I might add that she shows no differential activation for objects in area LO, obviously, because it's not there, and nor anywhere else in the ventral stream in a convincing way when you use line drawings where form is the only avenue to understanding what it is. So fMRI evidence then supports the notion. There is a lot of work, single unit work in monkeys that converges on this idea, and there's more controversial work in visual motor psychophysics about dissociations between the effects of size contrast illusions, for example, on grip amplitude versus perceptual judgments. Anyway, I'd like to spend a bit of time talking about some of the misconceptions that people have about this story, about the two visual systems. And the first is, of course, and it's worth emphasizing again and again, that we weren't the first to suggest that there's two visual streams in the primate cerebral cortex, as some people seem to think. Of course, that was Leslie Ungerleider and Mort Mishkin, who wrote a seminal paper in 1982, a chapter in, ironically, a book that I helped edit that was led by David Ingle, was out of a conference out of Brandeis. Of course, their work stems from Gordon Holmes' work, who was a leading British neurologist who studied young men from the First World War with penetrating missile wounds to parietal cortex, who suggested that they suffered from deficits in spatial vision. And you can see that echoing over the decades in Mort and Leslie's account of the where and what pathways. But I think our contribution was to emphasize the role of the visual streams in the control of action. And as you can see from this quote from our original monograph, we were really taking a kind of evo-devo approach, an evolutionary approach, arguing that natural selection really operates the level of behavior. In other words, somehow, unless we change the world in some way, natural selection has nothing to operate on. We've got to have some product for natural selection to distinguish, because being a brain in a vat and having great thoughts, unless it changes the world, is not going to provide any differential selection in the natural world. So we've argued that these two systems then evolved, one being indirectly linked to action, a perceptual system via cognition, and another which is very intimately linked with motor control. And indeed, this can be seen in this summary brain diagram from Jason Gallivan and Jody Cullum in Current Opinion, which they've mapped out over sort of a meta-analysis of many studies, areas that have been identified in the dorsal stream and related cortical motor areas. And, it was work by people particularly studying the saccadic system that pointed out that the systems involved in the control of eye movements in the posterior parietal cortex are intimately linked with corresponding structures in premotor cortex and, of course, the colliculus and brainstem. And beautiful work, I think to some degree ignored by Mitch Glickstein, has shown that these dorsal stream areas in the posterior parietal cortex are connected to the cerebellum, very heavily connected by way of relays in the pontine nuclei, the dorsolateral pontine nuclei. And there's a massive input from these visual areas. And as he points out, by contrast, there are few or no projections from cortical areas in the ventral stream. It looks like the dorsal stream is the conduit to get to these premotor and motor neurons in the brainstem into premotor and motor cortex in the frontal lobe. If you look at the ventral stream, and this is a summary of a whole host of studies led out of NIH by Leslie and Mort, and more laterally by Dwight Kravitz and his colleagues, that this network, the ventral stream, is really, as I said earlier, contacting the motor system through these connections that do a lot of cognitive processing, areas that are important for habit formation, for emotional processing, long and short-term memory, reward and value, and so on. So, just to return to the original question, you can ask, why on earth do we have two cortical visual systems? Why couldn't we have just one general purpose representation of the world that does it for everything we do, for thinking about the world and for acting? And I think the answer to that question, like the answer to so many questions, lies here. And you laugh because, of course, you recognize that as a glass of beer. And if you were actually in the scene and not looking at a photograph and you backed up from it, you'd still recognize it as a glass of beer. And if you squatted down and peered at it, it would still look like a glass of Canadian beer. And if you lean forward, it would look like beer. And indeed, if you have too many beers, the last thing you might see would be a glass of beer from a very different view, but you'd know that it was a glass of beer. All of that object constancy, of course, that's an old chestnut, and it's true that our perceptual system cares about constancies. The fact that I move around and there's huge geometric changes on my retina is largely ignored, and what I extract is information about the constant representation of the world. But that won't work for action. It'll let you watch television, but it won't let you act on the world. You have to have an understanding of the world. Your brain has to have an understanding of the world that's built in real-world metrics, or at least has a lot of fancy lookup tables over a lifetime of experience in order to get your hand on the object. So it's a very different kind of computation that's required for action versus perceiving the world, and so therefore different structures have emerged. I mean, what we're trying to say here then is that the visual information that's used by the dorsal stream for programming actions and for online control is not really perceptual in nature. And the important thing is it's not just unconscious, it's inaccessible to consciousness. It's somehow, we're conscious that we're acting and we're using visual information to act, and we may or may not be conscious of that particular action, but we're not in any way able to access the visual information that's being allowing us to hit the ball on the tennis court on the sweet spot of the racket, even though we know we're playing tennis. Larry, in fact, once characterized, back in 1997, he characterized the dorsal stream vision, as David and I were painting it, is in a sense, and this I think speaks to Larry's sense of humor, blind sight without blindness, in the sense that we as seeing individuals are blind to the information that is controlling many of our movements. So, David and I have argued then that dorsal stream vision and blind sight really stand in contrast to the visual processing in the ventral stream, even when that processing fails to reach awareness in the ventral stream. So, the ventral stream allows conscious or unconscious percepts. We can have perceptual information, as we've defined it at least, that we're unconscious of. Masked stimuli can influence our later behavior, stuff that we don't see at the time. And the dorsal stream, we can be conscious or not of the actions we perform. I just think it's kind of interesting that the product, as it were, of the ventral stream is something that is kind of visual by nature in the English language, and so we talk about it as vision, whereas the product of the dorsal stream transformations is an action, and the visual aspects of the control of that action are not privy to conscious scrutiny. Now, it's important to note that they're not hermetically sealed from one another. They both influence behavior, and they have to do this in a harmonious and non-competitive way. How am I doing for time? I know I started late, but okay. I'll get through this next section, and then I'm going to stop. Okay. So they don't work in isolation. They have to cooperate. In fact, for the ventral stream to be there, it's going to have to have influenced action in some fashion. Again, for the argument, it has to have something. Natural selection has to have something to operate on. So let's examine that in one context, the contribution of the two streams to the act of reaching out and grasping an object. This woman is not taking a selfie, I want to point out. She's actually picking some grapes, for those of you who can't really see that. So the dorsal stream, I would submit, can compute the sort of size of an object, its overall outline, its location, its orientation, without any contribution from the ventral stream. And we know that because patients with V1 lesions and patients with ventral stream lesions are able to act on objects using this information. But there are other kinds of products of grasping that after we make contact that have to do with the forces that we apply, for example, that only the ventral stream can provide information about. For example, the object's mass, how soft or hard it is, how fragile it is, what its friction is, what its temperature is, what its function is. And that's because we've learned associations between the visual appearance of objects and other properties that they have. So we learn about their material properties, we learn about their function through this associative learning. And so, you can look at functional graphs, for example. So if you present a screwdriver to someone like this with the business end facing them, then they will use this awkward grasp automatically. If you say, pick it up, they turn their hand very awkwardly and pick it up because they have end state comfort now, where they're ready to use the screwdriver. What they don't do is pick it up using a more efficient grasp, if you like, because now, unless they're going to stab somebody, it's not really in a state ready for that to happen. And so if you interfere with this behavior using a semantically demanding task that presumably engages those cognitive buffers that the ventral stream deals with in order to contact the motor system, then you find undergraduates, if they're doing this in parallel, use efficient grasp but don't show end-state comfort. And if you test DF on this with easy and awkward grasp demands with tools that she does not recognize, of course. She doesn't recognize tools. If you present them in the easy orientation, then she has end-state comfort-like controls on most of the trials because she just uses that efficient grasp, and that works when it's in the easy orientation. But when it's in the awkward orientation, now, as David Carey and David Milner showed, DF has real problems, and she often picks it up because she doesn't recognize it as a screwdriver in the wrong way. And this isn't because she can't rotate her hand efficiently using other demands like the longitudinal axis of the stimulus. So, like control subjects, she varies the posture according to the aspect ratio of the stimulus, whether it's a square or whether it's a rectangle. And so this will be my last little bit. Most recently, we've been getting in then into the brain and looking at tool recognition and looking at the dynamic changes that occur in contact between the dorsal and ventral stream when people are looking at tools versus non-tools. And of course, the important thing about tools is that almost all of them are elongated, which has not been a fact recognized by many studies that have looked at tools. And so you have to use elongated tools and stubby tools and elongated non-tools and stubby non-tools and so on as a proper control. And when you do this and you identify areas, the usual candidates come up, areas like the superior parietal region, MTG, premotor cortex, for tools, and they're very left hemisphere oriented for tools versus non-tools, and they're much more bilateral for elongated objects versus stubby objects. And when you start doing functional connectivity analysis, what you can see is, and you seed the parietal cortex, what you can see is activation in MTG and LO when you have tools versus non-tools, but nothing when you're using elongated objects versus stubby objects, because they're treated very similarly. They're not tools. And if you do, I can't pretend to understand this. This is Carl Friston. I have a mathematics postdoc who claims that he can understand, she can understand Carl's stuff. And when we do DCM, looking at the connectivity between tool and non-tool areas, we find an increase in the reciprocal connectivity between parietal cortex and MTG for tools versus non-tools, but we find only an increase in connectivity between the posterior parietal cortex and the ventral stream MTG for elongated objects. So it looks as though then toolness and elongation are separate properties of objects, and there's a reciprocal modulation of the interconnections between them between ventral and dorsal stream when in the left hemisphere in particular, when people are processing tools. But there's no evidence for this reciprocal modulation for elongated objects in general. So I think in the interest of time, I'll stop there, and thank you for your kind attention.
Apparently the microphone disappeared.
Oh. Could you hear me while I was talking, I hope?
No.
Oh. Yeah, that'd be bad.
On the issue of the dorsal stream and to what extent you're aware, you were very careful to say visually aware. But if I, as a child, am learning to play the piano and I'm reading music, I'm using a visual motor transformation. And of course, when I learn it, I am aware, but with automation, I'm not.
Yes.
So the question is, to what extent is it because you're looking at automated things that you're not finding awareness? And secondly, could you expand on what you mean by visual awareness?
Right. Yeah. So I'll deal with the last part first and then go back to piano playing. So perhaps we could use the example, as I used very briefly, about playing tennis, where you're engaged in a very rapid game of tennis. You're a good player with someone on the other side of the net, and the fact that your brain is calculating the tau of the tennis ball as it's coming towards you and is doing other kinds of predictions on the basis of visual information that your retina is processing and is delivering up to your posterior parietal cortex. You have no access to that information. You have access to the information. You made a pretty good shot on that occasion, but not of any. You have no awareness of the visual information that's being used. You are aware of the ball coming towards you, but I would argue that that's ventral stream access to MT's product and so on. It's not what's happening where MT is conveying that motion information up to the dorsal stream. That's what I mean by visual awareness. I don't know whether it's any clearer now. The thing about piano playing, yeah, David and I have thought about that. I keep invoking David's name not only to share the fact that he and I have worked very closely together on this, but also any weaknesses in the ideas. David and I have thought about the problem, not the problem, but the example of piano playing, driving a car, doing a lot of things that we become expert at. And I think, as many people have suggested, the circuitry becomes different. I think after practice, there is some change in the circuitry that is engaged.
It goes to parietal. That's been shown.
Yeah.
For learning to.
Yeah.
So what I'm saying
is... But what I'm saying is that the invocation of ventral stream and the cognitive apparatus with which it's associated in processing the musical score and delivering up the finger movements, I think some of that cognitive supervision drops out of the story and becomes more automated. And I think to some degree, the fact that we've developed skilled movements, the ability to make skilled movements, is why the posterior parietal cortex is developed, because otherwise we could do it with circuitry involving subcortical and cerebellar stuff, just like the frog, and we wouldn't have to have all of that apparatus of those reciprocal connections in the premotor dorsal stream network.
One more question?
So I'd like to ask you, Mel, about action.
Yeah.
I argue the ventral visual system is also involved in action.
Of course it is.
So,
yeah.
Exactly. So I have to recognize an object, perhaps compute its reward value,
yes.
and then I use something like perhaps my cingulate system to do action outcome learning, where the outcome is the reward, or whatever.
So there's action.
And here's what I wanted to ask.
Yeah.
So are the actions different? Are we dealing with instrumental actions with the output of the what system in general, and more sort of automated actions for the dorsal visual system, such as just reaching and angling your hand correctly?
Well, I think, for example, making an action of pressing the right button instead of the left-hand button in a task where there's reward or something else involved, that clearly engages the ventral stream and lots of cognitive buffers to solve the problem and make the decision. And you can call pressing the left-hand button an action, and I don't have any problem with higher order cognitive areas of ventral stream being involved in that, if that's what we mean by action. But if by action we mean directing our finger to the button and pressing it, which is what I mean right now when I'm talking about the posterior parietal cortex, then I think that is something that, for example, an optic ataxic patient might be able to recognize the solution to the problem but find it difficult to get her hand on the right button.
So I suspected that, Mel. So I think there's just a difference in the fields. So those who work on the ventral visual system and reward are then thinking of arbitrary instrumental actions.
Sure.
And when we refer to an action, that's what we mean,
Sure.
an arbitrary instrumental
Yeah,
action.
it's the taxonomy problem of taking the English language and mapping it onto brain.
It's just worth noting that.
Yeah, no, I completely agree. It's difficult. Yeah.
All right, so
Oh,
thank you, Mel.